Welcome back, everyone, to week 8 of Essentials in Hematology. Settle in, grab your coffee, because today is a big day. Today we are diving headfirst into hemolytic anemias and RBC structural disorders. Up until this point in the semester, we've focused heavily on recognizing patterns. You've learned how to look at RBC indices, identify normal and abnormal morphology, and classify anemias based on those numbers. You know what a microcytic hypochromic anemia looks like on paper and under the scope. But today, we shift gears. We are moving from simply recognizing a pattern to understanding the underlying process. We want to know not just what we see, but why we see it. Why did that cell take on that specific shape? What forces, either inside the cell or outside in the bloodstream, caused it to break down prematurely? Now, as upper-level histotech students, you need to be detectives, not just observers. You need to connect the visual evidence on the slide to the physiological chaos happening in the patient's body. So, on moving from pattern to process. Let's take a quick look at our roadmap and see where we are in the course. You've all done a fantastic job mastering the basics. You've conquered RBC indices such as MCV, MCH, and MCHC, and you know how to use them to categorize anemias. You've spent hours at the microscope identifying normal morphology and spotting the classic abnormal shapes. I hope so anyway. If not, you've had that luxury through the lectures and stuff I've presented. You've got the what down pat, but look at the right side of the timeline. This is where the real fun begins. We are now shifting our focus entirely to red blood cell destruction and mechanism-based thinking. We are going to look at what happens when red blood cells don't get to live out their peaceful 120-day retirement. We're going to look at cells that are built wrong from the start and cells that are perfectly fine but get thrown into a hostile environment. This is where hematology becomes a dynamic story of survival and destruction rather than just a static list of terms to memorize. This slide represents the most important mental shift you will make in this course. As histotechnologists, it is incredibly easy to get caught up in the visual identification game. You look in the scope and say, oh, look, a spherocyte. Next field. But I want you to push yourselves further. I want you to stop asking just, what do I see? And instead, I want you to look at that spherocyte and ask, what happened to this cell to make it look like that? Why did it lose its biconcave shape? Was it a genetic flaw in its scaffolding? Or did a macrophage take a bite out of it? And where in the body is this destruction taking place? Is it happening right there in the blood vessel? Or is it getting trapped in the spleen? Answering these why and where questions is the absolute key to truly understanding hemolytic anemias and providing meaningful diagnostic information to the pathologist. So let's start with the basics. What exactly is hemolysis? Simply put, it is the premature destruction of red blood cells. Normally, a red blood cell has a pretty good run. It gets pumped out of the bone marrow and circulates through the body, delivering oxygen. And after about 120 days, it gets old, it loses its flexibility, and then is quietly and efficiently retired by the macrophages in the spleen. It's a peaceful end. But in hemolysis, this survival time is significantly shortened. We're talking days or even hours instead of months. The cells are being destroyed long before their time. And when this happens, the body has to scramble to keep up. The bone marrow has to kick into overdrive to replace the lost cells, and the liver has to work overtime to clean up the mess of the broken hemoglobin. Hemolysis isn't just a low red count, it's a state of high turnover chaos. Whenever you are faced with a patient who has anemia, meaning a low hemoglobin and hematocrit, the very first fundamental question you must always ask yourself is this: is this a problem with production, or is it a problem with destruction? Think of the bloodstream like a bathtub. If the water level is low, as in anemia, there are only two reasons why. Either the faucet is turned off, which is a production problem in the bone marrow, or the drain is wide open, which is destruction or blood loss. Is the bone marrow failing to make enough cells because it lacks iron, B12, or is it just diseased? Or are the cells being made perfectly fine, but then they are being destroyed prematurely or lost through bleeding? This distinction is crucial because it completely dictates your entire diagnostic approach in the tests you will run next. So how do we answer that question? How do we know if the faucet is on or off? We look at the marrow's response, and we do that by checking the reticulocyte count. The reticulocyte count is your direct window into the bone marrow's factory floor. If a patient is anemic and the reticulocyte count is significantly increased, it tells you the bone marrow is healthy. It recognizes the anemia and it is working overtime, pushing out immature red blood cells to compensate for destruction or loss. The factory is running at 110% capacity. However, if the patient is anemic and the reticulocyte count is decreased, or even just normal, you have a production problem. A normal retic count in the face of severe anemia is actually abnormal. It means the marrow isn't responding to the crisis, and the factory is broken. Okay, so let's say the retic count is high, and we've established that hemolysis is indeed occurring. The next big question is, where is the crime scene? Where is this destruction happening? There are two major types of hemolysis based on location: intravascular and extravascular. Intravascular means exactly what it sounds like. The destruction is happening right there inside the blood vessels, in the general circulation. Extravascular means the destruction is happening outside the main blood vessels, primarily in the tissues of the spleen and the liver. Understanding the difference between these two locations is absolutely vital, because they produce very different clinical symptoms and very different laboratory results. So let's break them down. Let's look closer at intravascular hemolysis. This is the dramatic one. This occurs directly in the circulation. The red blood cells literally rupture or explode within the bloodstream. Imagine a water balloon popping inside a pipe. It makes a mess right there in the flow. When these cells burst, they release all their internal contents, most notably massive amounts of free hemoglobin directly into the plasma. This is usually a more acute, severe, and sometimes life-threatening process. The body is not designed to handle large amounts of free hemoglobin floating around in the blood. It's toxic to the kidneys and can cause serious complications. We see this in severe transfusion reactions, certain infections, and mechanical trauma to the cells. Extravascular hemolysis, on the other hand, is a bit more stealthy. It occurs primarily in the spleen and the liver. Here, abnormal, damaged, or antibody-coated red blood cells are recognized by the body's security guards, the macrophages. It's a more controlled demolition. The macrophage engulfs the red blood cell and breaks it down internally, safely containing that hemoglobin. This is actually the most common form of hemolysis. In fact, it's exactly how normal old red blood cells are cleared every single day. The problem in extravascular hemolytic anemia is just that this normal process is happening way too fast, and perfectly good or slightly flawed cells are being targeted prematurely. The spleen becomes a very busy and often very enlarged organ. Let's compare the two side by side, because you need to be able to distinguish them based on lab results and clinical signs. Intravascular hemolysis often presents acutely. Because free hemoglobin is dumped into the blood, it gets filtered by the kidneys, leading to hemoglobinuria, that classic root beer or cola colored urine. Morphologically, you'll often see schistiocytes or fragmented cells on the smear because the cells are being physically torn apart in the vessels. Extravascular hemolysis is often a more chronic, ongoing process. Because the macrophages in the spleen are working overtime eating cells, the spleen physically grows, leading to splenomegaly or splenomegaly. You won't see hemoglobin in the urine because the macrophages contain it. And the classic morphological finding here is the spherocyte, which we'll discuss in detail shortly. Now that we've talked about where hemolysis can happen, let's dive into how we actually prove it's occurring in the lab. We're looking for specific chemical footprints that tell us red blood cells are being destroyed prematurely. Think of these as the body's alarm bells, each ringing for a slightly different reason. We'll focus on 4 key markers: reticulocytes, bilirubin, LDH, and haptoglobin. Each marker gives us a piece of the puzzle, helping us understand the extent and even the type of hemolysis. It's like forensic science for your blood cells. Let's break down what each of these markers means. First up, reticulocytes, which are essentially the bone marrow's urgent message for help. When red blood cells are being destroyed faster than normal, the bone marrow kicks into overdrive, trying to compensate for the loss. It starts pushing out immature red blood cells, or reticulocytes, into the circulation. On a standard right-stained smear, you'll see these as larger, bluish cells, a phenomenon we call polychromasia, which is due to residual RNA. So if you see a lot of polychromasia, it's a strong indicator that the marrow is working hard to replace lost cells, which is a classic sign of hemolysis. But remember, an increased reticulocyte count only tells us the marrow is responding. It doesn't tell us why. Next, we'll look at what happens when hemoglobin itself breaks down. Following the reticulocyte response, we turn our attention to bilirubin, which is the direct breakdown product of hemoglobin. When red blood cells are destroyed, the hemoglobin inside them is released and then processed. The heme portion of hemoglobin gets converted into unconjugated bilirubin. If there's a lot of hemolysis, there's a lot of hemoglobin breakdown, and that means a big surge in unconjugated bilirubin in the blood. Clinically, this often shows up as jaundice, that telltale yellowing of the skin and eyes, a clear sign that the body is struggling to process all that breakdown product. It's a vivid indicator that red blood cells are being destroyed too quickly. Now, let's talk about the time that when cells burst. Moving on from bilirubin, we have lactate dehydrogenase, or LDH, which is an enzyme that's usually tucked away inside red blood cells. Think of LDH as the internal contents of the red blood cell. When a red blood cell is destroyed, whether it's bursting in the bloodstream or being eaten by a macrophage, its contents spill out into the plasma. This includes LDH. So if you see elevated serum LDH levels, it's a very sensitive indicator that cells are being damaged and releasing their insides. It's not entirely specific to hemolysis, as other conditions can also raise LDH, but in the context of other markers, it's a strong piece of evidence. If cells are popping, LDH is rising. And next we'll look at the body's cleanup crew for free hemoglobin. Finally, we come to haptoglobin, which is like the body's dedicated scavenger for free hemoglobin. Haptoglobin is a protein made by the liver, and its sole purpose is to bind to any free hemoglobin floating around in the plasma. This binding prevents the hemoglobin from causing damage and also allows the liver to clear the complex. In cases of intravascular hemolysis, where red blood cells burst directly in the bloodstream, massive amounts of free hemoglobin are released. Haptoglobin rushes in to bind it all up. And because it gets used up so quickly, its levels in the serum drop significantly, often becoming undetectable. This makes a decreased haptoglobin level a really important indicator, especially for intravascular destruction. Now let's put all these pieces together to see the full picture. So we've talked about the individual lab markers, but how do they all fit together? When you see this specific combination of lab results, you can confidently say, we have hemolysis here. This profile is your go-to for confirming red blood cell destruction. We're looking for increased reticulocytes because the bone marrow is working overtime to replace lost cells. You'll also see elevated LDH and unconjugated bilirubin. Both are direct consequences of red cell breakdown. And critically, haptoglobin levels will be low, especially if the destruction is happening right in the bloodstream. If you spot this pattern, you've nailed the diagnosis of hemolysis. Now, the real detective work begins, figuring out why it's happening. Once we've confirmed hemolysis with that lab profile, the next crucial step is to determine the source of the problem. Is it intrinsic or extrinsic? This distinction is fundamental to understanding the underlying pathology. Think of it this way. Is the red blood cell itself flawed from the start? Or is it a perfectly good cell being attacked by something in its environment? This isn't just academic. It completely changes your diagnostic pathway and treatment strategy. It's the difference between a manufacturing defect and external damage. So let's start with intrinsic causes. These are almost always hereditary, meaning the red blood cell was literally built to fail due to a genetic defect. We're talking about issues with the cell's fundamental components. We can broadly categorize these into three main areas. Membrane defects, this is where the cell's outer structure is compromised. Enzyme deficiencies, this is where the cell lacks vital metabolic machinery. And hemoglobinopathies, where the very oxygen-carrying protein is malformed. These are all problems originating from within the cell itself, making it inherently fragile or dysfunctional. Now, shifting gears from intrinsic problems, we look at extrinsic causes. Here, the red blood cell is perfectly healthy and functional, but it's operating in a hostile environment that's actively damaging or destroying it. These causes are typically acquired, not inherited. We'll explore three major categories. Immune-mediated destruction. This is where the body's own defenses mistakenly target red cells. Mechanical hemolysis. This is where physical forces literally tear the cells apart. And then there's infectious causes, which is where pathogens invade and destroy the red blood cells. Think it's like a well-built car getting into an accident, rather than breaking down due to a faulty engine. From our discussion of intrinsic causes, let's dive deeper into membrane disorders, starting with the unsung hero, the red blood cell membrane. This isn't just a simple bag holding hemoglobin. It's an incredibly complex, dynamic structure. It relies on a sophisticated network of proteins like spectrum and ankyrin to maintain its unique biconcave shape and provide the extreme flexibility needed to squeeze through tiny capillaries. Without this flexibility, the cell can't do its job. And because it certainly can't survive its 120-day journey, any defect can lead to premature destruction, as we'll see with hereditary spherocytosis. So what happens when that complex membrane structure we just discussed starts to fall apart? The most common culprit is hereditary spherocytosis, where the red blood cell literally starts losing bits of itself. This isn't just some random shedding. It's a direct consequence of genetic mutations in those crucial vertical membrane proteins, like spectrin and ankyrin, that anchor the lipid bilayer to the underlying cytoskeleton. Imagine a tent with weak guy ropes. It can't hold its shape. As the cell loses surface area but keeps its volume, it's forced into the most compact shape possible, a sphere. This loss of the biconcave disc shape has huge implications for the cell's function and survival, setting it up for premature destruction. Now, how do we actually spot these little troublemakers on a smear? When you're looking at a peripheral smear, spotting a spherocyte isn't just about identifying a round cell. It's about recognizing its distinct characteristics. These cells appear smaller and much denser than your average red blood cell. The key giveaway? They lack the central pallor that's characteristic of a normal biconcave disc. Think of it like a perfectly inflated basketball versus a deflated one. The basketball is uniformly dense. This density isn't just visual, it's reflected in the mean corpuscular hemoglobin, or MCHC. Mean corpuscular hemoglobin concentration, or MCHC, which is often elevated, sometimes even above 36 grams per deciliter, making it one of the few conditions where you'll see that. But why is this spherical shape such a problem for the red blood cell? So we've identified the spherocyte, but why does its spherical shape lead to its demise? It all comes down to flexibility, or rather, the lack thereof. Spherocytes are rigid. They've lost that incredible deformability that allows normal red blood cells to squeeze through the tightest capillaries. When these rigid spherocytes try to navigate the narrow, tortuous pathways of the splenic cords, they get stuck. They can't contort themselves to fit. Once trapped, the ever-vigilant splenic macrophages recognize them as abnormal and quickly remove them from circulation. This is a classic example of extravascular hemolysis, and it's why splenomegaly is a common finding in hereditary spherocytosis. But spherocytes aren't the only oddly shaped cells we see from membrane defects. While spherocytes are losing ground, another membrane defect, hereditary elliptocytosis, is stretching the limits of red blood cell shape. Unlike HS, where vertical proteins are the issue, HE involves defects in the horizontal protein linkages of the membrane skeleton. This means the cells can stretch out into an elongated or elliptical shape as they pass through capillaries, but they can't quite snap back to their normal discoid form. You'll see these cigar-shaped cells all over the smear. Interestingly, even though the morphology looks dramatic, the actual hemolysis in HE is often much milder than in spherocytosis, and many patients live completely asymptomatic lives. Now, let's shift gears and look at another intrinsic problem, enzyme defects. We've talked about membrane defects, but now let's dive into enzyme deficiencies, which are another major intrinsic problem for red blood cells. Why do these cells, which seem so simple, need such a robust enzyme system? Well, remember, mature red blood cells are unique. They've kicked out their nucleus and mitochondria. This means they can't synthesize new proteins or repair themselves. Basically, they're entirely dependent on their pre-existing enzyme machinery to generate energy and, critically, to protect themselves from constant oxidative damage. If an enzyme is deficient, that cell is essentially flying without a shield, making it incredibly vulnerable to stress. The most common of these enzyme defects is G6PD deficiency, which we'll explore next. So let's kick off our discussion of enzyme defects with the big one, glucose 6-phosphate dehydrogenase, or G6PD deficiency. This is the most common red blood cell enzymopathy globally, and it's a classic example of how a seemingly small genetic flaw can lead to big problems when the cell is stressed. Remember how we talked about enzymes being the cell's defense system? Well, G6PD is a key player in that defense, specifically against oxidative stress. Patients with G6PD deficiency are usually perfectly fine until they encounter an oxidative trigger. This could be certain drugs like some antimalarials, or a severe infection, or even something as seemingly innocuous as eating fava beans. That's right, fava beans can trigger a hemolytic crisis in these individuals. It's a fascinating example of gene-environment interaction. Now let's see what happens inside the cell when that oxidative stress hits. When a G6PD deficient cell faces oxidative stress, it's like a superhero without its powers. It just can't neutralize the threat. The oxidative agents go to town on the hemoglobin molecules, causing them to denature and precipitate. Think of it like cooking an egg. The proteins change structure and clump together. These clumps of denatured hemoglobin don't just float around. They actually attach to the inner surface of the red blood cell membrane. We call these sticky, denatured hemoglobin clumps Heinz bodies. They're a direct result of the cell's inability to protect itself from oxidative damage. And these Heinz bodies are going to cause some serious trouble for our poor red blood cells, as we'll see next. Now, about these Heinz bodies we just discussed. You can't just spot them on a regular right stain smear. And that's a crucial point for you as histotechs. To visualize Heinz bodies, you need a special technique called a supravital stain, like new methylene blue. This stain allows you to see inclusions within living or recently living cells. When you use a supravital stain, Heinz bodies pop out as dark blue or purple inclusions, typically clinging to the right edge of the cell membrane. They're like little barnacles on the inside of the red blood cell. So, if you suspect G6PD deficiency, remember to reach for that supravital stain. But what happens to these cells once they have these peculiar inclusions? So what happens to a red blood cell that's carrying around a Heinz body? As this compromised cell tries to squeeze through the narrow passages of the spleen, the resident macrophages are on patrol. They recognize that something is off with this cell, specifically that abnormal inclusion. The macrophage will literally take a bite out of the red blood cell, attempting to remove the Heinz body. And when it does, it takes a chunk of the cell membrane right along with it. The result? A cell that, on a right stain, looks like an apple with a bite taken out of it. We call these bite cells, or degmacytes. It's the spleen's way of trying to clean up the mess, but it leaves the red blood cell permanently disfigured. And sometimes we see the stage right before the bite. Before the spleen takes its full bite, you might sometimes catch the red blood cell in an intermediate stage. The membrane around the damaged area, where the Heinz body is, can fuse together, creating a clear vacuole or a blister at the edge of the cell. These are what we call blister cells. They're essentially the precursors to those bite cells we just talked about. Seeing both bite cells and blister cells on a peripheral smear is a really strong indicator of oxidative damage and should immediately make you think about G6PD deficiency. It tells you the cell has been under attack and the spleen has been trying to intervene. Next, we'll shift gears to another intrinsic problem, issues with the hemoglobin itself. Now we shift gears to our third category of intrinsic defects, hemoglobinopathies. These aren't about the cell's wrapper or its internal machinery, but about the very core of its identity, the hemoglobin molecule itself. Remember, a red blood cell is essentially a bag of hemoglobin, and if that hemoglobin is structurally flawed, the entire cell's integrity is compromised. This means the cell's shape, its ability to carry oxygen, and its flexibility are all at risk. A defect here is a fundamental flaw in the cell's purpose. We're talking about qualitative defects where the body makes enough hemoglobin, but it's just not the right kind. This is a big one, so let's dive into the classic example. The poster child for hemoglobinopathies, and the one you absolutely must know, is sickle cell disease. This isn't some complex multi-gene mutation. It's a single tiny change in the genetic code. Just one amino acid substitution, valine instead of glutamic acid, at a specific spot on the beta globin chain. But this seemingly small change has massive consequences. Under low oxygen conditions, this abnormal hemoglobin S doesn't just sit there. It polymerizes, forming long, rigid rods inside the red blood cell. Imagine trying to fold a perfectly good tent versus one with rigid, unbending poles. That's what's happening inside these cells. This polymerization is the root cause of all the trouble, and it dramatically alters the cell's behavior. This polymerization of hemoglobin S literally twists and distorts the red blood cell into that unmistakable crescent or boat shape that we call a drepanocyte or sickle cell. These cells are no longer the flexible biconcave discs designed to glide through the capillaries. They're hard, stiff, and pointed. Initially, this sickling can be reversible if oxygen levels return to normal. But with repeated cycles, the cell membrane becomes permanently damaged. Once that happens, the cell is irreversibly sickled, and its fate is sealed. These cells are a dead giveaway on a peripheral smear, and they tell a very clear story of what's happening at the molecular level. The clinical impact of these rigid, sickled cells is profound and devastating. First, these misshapen cells are rapidly recognized as abnormal and destroyed by the spleen, leading to chronic extravascular hemolysis and severe anemia. But the real kicker, the thing that causes so much pain and organ damage, is vasoocclusion. These stiff, sticky sickle cells get jammed in the tiny capillaries, blocking blood flow. This leads to excruciating pain crises and, over time, causes ischemic damage to virtually every organ in the body. It's a cascade of problems, all stemming from that single amino acid change. It's a stark reminder of how a microscopic flaw can have macroscopic consequences. In sickle cell disease and many other hemoglobinopathies, you'll often spot target cells, also known as podocytes. These cells have that characteristic bullseye appearance, and they form when there's an excess of membrane surface area relative to the cell's internal volume. Think of it like a deflated basketball. The skin is there still, but the volume is reduced, making it look like a target. While they're a common finding in hemoglobinopathies and thalassemias, You'll also see them in iron deficiency and liver disease. So while they're not specific to sickle cell, seeing them should always make you consider a problem with hemoglobin or membrane to volume ratio. Now, let's shift our focus to extrinsic causes of hemolysis, where the problem isn't with the red blood cell itself, but with its environment. We've talked about intrinsic problems, but now let's shift to extrinsic causes, starting with immune hemolysis. This is a classic case of friendly fire, where the body's own defense system mistakenly attacks its red blood cells. Once these red blood cells are coated in antibodies, they become targets for destruction by macrophages, primarily in the spleen and the liver. It's like a misidentified target in a battle, leading to a collateral damage. This process is a major cause of extravascular hemolysis, as the cells are removed by the reticuloendothelial system. But how do we actually prove this immune involvement? To confirm that immune mechanisms are at play, we rely on the direct antiglobulin test, or DAT. This test is incredibly powerful because it directly detects antibodies or complement components already stuck in the patient's red blood cells while they are still inside the body. A positive DAT is the smoking gun, unequivocally confirming that an immune process is actively destroying those red cells. It's a critical step in differentiating immune hemolytic anemia from other causes. And it tells us that the problem isn't with the cell itself, but with something attacking it from the outside. But immune attacks aren't the only external threat to red blood cells. Beyond immune attacks, red blood cells can also face purely physical destruction, which we call mechanical hemolysis. The most common and clinically significant form is microangiopathic hemolytic anemia, or MAHA. Imagine a tiny obstacle course in your blood vessels, created by microthrombi, which are small clots that partially block the flow. As red blood cells are forced through these constricted, fibrin-laden pathways at high speed, they are literally torn apart. It's like trying to push a delicate object through a shredder. The cells just can't withstand the shearing forces. This physical trauma leaves behind a very specific morphological clue. What do these shredded cells look like? The telltale sign of mechanical hemolysis, especially MAHA, is the presence of schistiocytes on the peripheral smear. These are fragmented red blood cells, often appearing as helmet cells, triangles, or other bizarre shapes, always with sharp, irregular edges. Seeing schistiocytes is a critical finding that demands immediate attention, as it points to a microangiopathic process that can be life-threatening. These fragments are direct evidence of the physical trauma the cells have endured in the circulation. But mechanical forces aren't the only other hostile environments red blood cells can encounter. Beyond immune and microangiopathic causes, red blood cells can face other hostile environments. For instance, prosthetic heart valves can cause macroangiopathic hemolysis, where the sheer force of blood flow around the artificial valve physically smashes the cells. Then there are infections, like malaria or babsiosis, where parasites invade and multiply within the red blood cells, eventually rupturing them. Even severe thermal injuries, like extensive burns, can directly damage the red blood cell membrane, leading to fragmentation and the formation of microspherocytes. These diverse external factors all highlight how vulnerable red blood cells are to their surroundings. But regardless of the cause, where do most of these damaged cells end up? After discussing all those hostile environments, let's take a moment to really appreciate the spleen. This organ is truly the ultimate filter, tirelessly inspecting every red blood cell that passes through its intricate network. Its primary job is to identify and remove any red blood cells that are old, rigid, or have been tagged by antibodies. This constant surveillance makes the spleen the major site of extravascular hemolysis, where macrophages are constantly at work. Because it's often working overtime in chronic hemolytic anemias, you'll frequently see splenomegaly or an enlarged spleen as a clinical sign. So while the spleen is a hero, it can also be a bit of a villain for abnormal red blood cells. Next, we'll look at the liver, another crucial player in the hemolytic process. While the spleen is the filter, the liver acts as the body's processing plant, handling the aftermath of all the red blood cell destruction. Its main role here is to take the unconjugated bilirubin, which is a toxic byproduct of hemoglobin breakdown, and conjugate it. This makes it water soluble, so it can be safely excreted in the bile. The liver also produces haptoglobin, that scavenger protein we talked about, and then clears the haptoglobin hemoglobin complexes that form during intravascular hemolysis. When the rate of hemolysis overwhelms the liver's capacity to process all this bilirubin, that's when we start to see jaundice. Now that we've covered the internal players, let's bring it all together with the clinical picture. So what does all this mean for the patient? When you put together the mechanisms of hemolysis, you start to see a classic clinical picture emerge. The hallmark triad of hemolysis includes anemia, which manifests as pallor and fatigue, jaundice due to those elevated bilirubin levels, and splenomegaly from the spleen working so hard. If the hemolysis is happening intravascularly, you'll also see dark hemoglobinuric urine, often described as root beer colored. Recognizing this triad is crucial for suspecting hemolysis at the bedside. With these clinical signs in mind, let's circle back to what you see under the microscope, because morphology tells you a story. We've covered a lot of ground today, and I want to reiterate a point that can't be stressed enough. Red blood cell morphology is never random. Every single abnormal shape you encounter on a peripheral blood smear is a physical record, a fingerprint of the stress or damage that cell has endured. It's not just a pretty picture, it's a direct reflection of the underlying pathophysiology. Your job as histotechnologists is to learn to read that record, to interpret the story each cell is trying to tell you. This is where pattern recognition becomes mechanism-based thinking. Let's do a quick review of some key patterns. Let's quickly review some of the key morphologic patterns we've discussed today, starting with spherocytes and schistiocytes. When you spot spherocytes, those dense, small cells lacking central pallor, immediately think membrane loss. This typically points to extravascular hemolysis, And your differential should include immune-mediated destruction, which you'd confirm with a DAT, or hereditary spherocytosis. On the other hand, if you see schistiocytes, those fragmented, jagged cells, your mind should jump to physical fragmentation. This is the hallmark of intravascular mechanical damage, and you must, absolutely must, rule out microangiopathic hemolytic anemia, or MAHA. These two patterns are critical for differentiating the type of hemolysis. Next, we'll look at bite cells and target cells. Building on our previous pattern recognition, let's now look at bite cells and target cells, which tell us about different types of cellular stress. When you see bite cells or blister cells, your mind should immediately go to oxidative damage and the spleen literally taking a bite out of the cell. This is the classic morphological clue for G6PD deficiency, where the cell can't handle oxidative stress and then forms Heinz bodies that the spleen then tries to remove. Conversely, target cells, or podocytes, signal an imbalance in the red blood cell surface area to volume ratio. This often points towards issues with hemoglobin itself, like in hemoglobinopathies or thalassemias, or it can be seen in liver disease. These patterns are your visual roadmap to the underlying pathology. Now let's consider another crucial visual clue that tells us about the bone marrow's efforts. While all those abnormal shapes tell us how red cells are dying, polychromasia gives us a vital piece of information about the bone marrow's response. It's trying. Polychromasia, those slightly larger bluish tinged cells, are essentially reticulocytes that the marrow is pushing out in a hurry. It's a sign of increased erythropoiesis, the body's attempt to compensate for the ongoing red cell destruction. If you see significant anemia but no polychromasia, that's a red flag. It means the bone marrow isn't responding, which points to a much more severe problem than just destruction. So, when you see polychromasia, you know the factory is working overtime. Understanding these patterns is key, but we also need a structured way to approach diagnosis. To avoid getting overwhelmed by all these patterns, we need a systematic approach to evaluating anemia. First, always confirm that anemia is actually present. Don't ever assume. Second, check the reticulocyte count to see if the bone marrow is responding appropriately to the low red cell count. An increased retic count points to destruction or loss, while a low or inappropriately normal count suggests a production problem. Third, if the marrow is responding, look for the chemical evidence of hemolysis, elevated bilirubin and LDH, and decreased haptoglobin. Finally, use the morphology from the peripheral smear in specific tests like the DAT to pinpoint the exact type and cause of hemolysis. This stepwise thinking ensures you don't miss critical information. And as you go through these steps, always keep one fundamental question in mind. As you work through a case, always return to this fundamental question. Is the problem intrinsic to the red blood cell, or is it extrinsic, meaning the environment is damaging it? This is your final check, your ultimate diagnostic filter. If the red blood cell was built wrong, you're looking at hereditary conditions like spherocytosis or G6PD deficiency or hemoglobinopathy. If the red blood cell was perfectly fine but got attacked or damaged, then you're thinking about immune hemolysis or mechanical destruction or even an infection. This distinction is paramount because it completely changes your diagnostic and treatment pathway. But remember, no single piece of information will give you the whole picture. You can't rely on just one piece of the puzzle. You need to put all the data together to get the complete picture. The CBC gives you the quantitative numbers. The peripheral smear provides the crucial visual evidence of what's happening to the cells. And the chemistry panel confirms the destruction and helps differentiate types of hemolysis. Add to the patient's clinical history and symptoms, and you start to see the full story. Synthesizing all this information is what makes you an effective histotechnologist. It's about connecting the dots from the lab bench to the patient's bedside. Now, let's talk about how to approach a case study with this comprehensive mindset. After synthesizing all that data, it's tempting to jump to conclusions, but resist that urge. When you're approaching a case, especially in an exam or clinical setting, you absolutely cannot jump straight to a diagnosis just because you spot one interesting cell. That's like reading one word in a book and thinking you know the whole story. Instead, you need to build your reasoning step by step, almost like a detective. First, confirm the anemia, then prove the hemolysis with the lab markers we discussed. Only after you've laid that groundwork can you start to formulate your differential diagnosis. This methodical approach ensures accuracy and prevents misdiagnosis. It's about showing your work, not just guessing the answer. This systematic thinking will serve you well as we summarize our key takeaways. So let's distill everything we've covered today into a few key takeaways. Remember, hemolysis isn't just a fancy word. It's the premature destruction of red blood cells, cutting short their vital 120-day journey. Every weird shape you see on that smear, the spherocyte, the schistiocyte, the bite cell, it's not random. It's a direct reflection of the underlying mechanism of destruction, a visual clue to the cellular trauma. And don't forget our unsung heroes, the spleen and the liver. They're the major players in clearing these damaged cells. Finally, always categorize the problem as intrinsic to the cell or extrinsic, because that distinction fundamentally changes your diagnostic path. These core concepts will be your compass in navigating hemolytic anemias. Now, let's reflect on how far you've come in your understanding. Building on these key takeaways, I really want to acknowledge your growth throughout this course. You've moved beyond simply identifying patterns, which is a great start, but now you're digging deeper. You're learning to understand the intricate mechanisms behind these diseases, asking not just what, but why. This shift in thinking, from mere observation to mechanistic understanding, is what evaluates you or elevates you from a good histotech to a truly great one. It's about critical thinking, not just root memorization. You're developing the diagnostic intuition that comes from understanding the how and why. With this new perspective, let's briefly look at what's coming next week. Now that we've thoroughly explored the world of red blood cell destruction, we're going to pivot next week. We spent today focusing on what happens when red blood cells are destroyed too quickly. But what happens when the factory itself, the bone marrow, isn't producing enough cells in the first place? Next week, we will flip the script entirely and delve into bone marrow failure and other production problems. It's a completely different set of challenges with its own unique set of morphological clues and diagnostic pathways. Get ready to shift your gears again as we tackle the other side of the anemia coin. But before we go, I have one final thought for you. I'll leave you with this final thought, a mantra to carry with you. Every single red blood cell on that slide, whether it's perfectly normal or wildly distorted, is trying to tell you a story. It's a narrative of its life, of its struggles, and of its demise. As histotechnologists, your profound responsibility is to listen intently to that story, to look closely at every detail, and to interpret it accurately. Your careful observation and understanding are crucial for patient care. Have a fantastic week, everyone, and I'll see you next time. Thanks.